So it is my pleasure to introduce today's speaker for our Grand Round series. Dr. Jeffrey Chuang is joining us from the Jackson Laboratory today. We were speaking just before the seminar today because actually my training is in biophysics, and it's not every day that I get to introduce a speaker who actually trained in hardcore physics. So that's like a real affinity for me, and I think it kind of reminds us in how much leveraging people who come to science and to medicine from different backgrounds is such an incredible way to have this alignment of brains and insights and kind of driving questions that ultimately is so important to our ability to improve the treatments for cancer. So I think it's like that Jeff is a great example of that. Yes, we started off at MIT doing a PhD in theoretical physics, including in DNA biophysics and polymer physics, which is super relevant for how we think about how the chromosomes behave inside living nuclei. And from there, he went to the West Coast to UCSF and did a computational biology postdoc with Hao Li. Came back to the East Coast initially to start his independent group at Boston College in the biology department. And then from there moved to the Jackson Laboratory, where he is now a professor. And the Jackson Laboratory, there's also these academic relationships also with UConn. And so he also holds appointments at UConn in genetics and genome sciences. So he is now the deputy director of the Jack's Cancer Center. And he continues to use computational approaches for, I think, one of the problems that we're all facing now, which is this incredible amount of big data and how we're actually able to connect this and leverage approaches like machine learning, not just for omics data, but all of the kinds of data, including tissue image analysis, which is something that's super interesting to me as a cell biologist, is something that resonates with me as well. And then also to how we can leverage patient-derived cancer models in ways that can improve ultimately our treatment of cancer. And his laboratory has been supported by the NIH, the NSF, the DoD, and the Hope Foundation, and are really excited to have him visiting us today. Thank you, Jeff.
Thank you so much, Megan. I really appreciate the invitation to come and speak. And really, it's such an honor to come here and talk with you all here. I don't often get a chance to talk in kind of a clinically kind of oriented audience here. It's actually my first time speaking in a grand round. So thank you again for this. I'm excited. You know, I work on problems in computational biology. I think some of what I'll talk about will be kind of wonky in terms of methods, but not too much. My feeling these days is that big challenge of our time is that the technology is going so fast with things like AI, also like new profiling technologies, that often, at least the way I think about it, is we want to find things that we can understand enough to do something with and develop treatments. Sometimes it's the algorithms themselves, but sometimes it's just getting a feel for what the data are telling us. So my lab really works at this intersection. We try to come at things with different techniques, some things biophysical because of my training, some things algorithmic, but really bringing in perspectives even from surgeons. We work with a lot of pathologists too, and of course, cancer biologists. So I think this particular visit, the genesis of it was some discussions between our cancer centers. And so I just want to mention a little bit about the Jack's Cancer Center. I hope many of you will take the opportunity to visit us sometime. And so our rubric is that we aim to discover precise genomic solutions to advance cancer medicine through basic genetics and biology. Some of you may be familiar with Jack's. I think we're most well known for our mouse genetics work. We actually work in many different areas. We have our own NCI supported cancer center. And we're one of 7 basic research centers supported by the NCI. So this is a little bit of a description of our program. If you look at the bottom here, kind of our historical noteworthiness is understanding cancer genetics, which comes from our expertise in mouse genetics. Again, I think many of you have probably bought mice from the Jackson lab. That's our kind of commercial side, but we have actually about 60 faculty spread across 2 campuses, and I'm one of those. Our current cancer center director is Carolina Paluca, who is a cancer immunologist. In previous years, we had been led by Edison Liu. You see him on the left here. And his expertise is really more about cancer genetics and understanding treatment resistance. But in the past couple of years, since Dr. Paluca has been our cancer center head, we've really tried to morph into understanding extrinsic factors that impact the tumor microenvironment and specifically the immune system and how it can modulate cancer treatment response. More broadly speaking, we've kind of interwoven this with the strengths of the Jack's Cancer Center community, thinking especially about aging in cancer, because again, as many of you know, aging is the biggest risk factor for cancer. And we have special projects that we can take on at the Jackson Lab in which we age mice and think about that aging in kind of a orthogonal way to how we can do it clinically. So I'll be telling you some of the stories that are related to those ideas in the talk today. These are just some of our members. I think we have a really broad audience of excellent researchers. Maybe some of you know or have friends among this group, but please come see us. Okay, so now for my talk, what I'm going to tell you most about today is image analysis, which is my lab's specialty. I started working on this about 6 years ago. Mostly, as Megan had mentioned, I had done more like genetics and gene regulation, and then actually tumor heterogeneity. But at that time, I became frustrated with looking at tumor heterogeneity from sequence data because I always felt that we didn't know what the spatial relationships of the samples were to one another. And at that time, there was this big wave, which was that at places like Google and Facebook, there was this wave of image data which were coming in, and there was a growth in kind of machine learning methods to analyze them. And kind of a little bit after that wave, there started to be a big amassing of tumor image data, including from all these H&E banks that hospitals manage. So the technique that most people have used to analyze image data over the last several years have been deep neural networks. For those of you who are not an expert in this field, they're a little bit different than what are done with like large language models, but have some similarities. This is kind of a core structure. And I think at this point, almost everybody has used a version of this where you have an image, they tell you if there's a cat in it, or are there people doing, ABC, jumping up and down, et cetera. This is kind of a standard thing now. And we can use these tools for image analysis in cancer. And I think that this field is just growing so fast, really in the same ways that people are using like chatbots and these tools from the big tech companies. The image analysis field is growing in the same way and in many ways even faster. So what do we want to do with these image analysis tools? One is we'd like to just directly predict health outcomes. So if I take the way pathologists always have. So if I have a picture of a tumor, whether it be H&E, whether it be some of these new multiplex technologies, can I tell you, number one, how is the patient going to do? Number two, can I tell you what treatment is going to work the best? The way that my lab thinks about these problems is we want to put some structure on kind of this kind of essentially pathological analysis. So we want to identify recurrent spatial structures within these images. And our goals are, number one, to simplify these very complex spatial behaviors into interpretable features. Number two, to distinguish targets for treatment development. And I think that depends also on this interpretation. We need to have some understanding in order to feel confident and also be able to convey that to patients eventually when treatment recommendations are made. And then third, to elucidate biophysical factors that may affect targetability. So things like, is there a strong fibrotic behavior between, the bloodstream and then, where most of the tumor is. Those are things that are not so obvious when you're developing things from a genetics perspective, but are actually quite important for true targetability. So first, I want to tell you just a little bit of background, because I know that the audience for these kinds of topics is very diverse, and the field is moving so fast. So this is actually a little bit of an old story now. This is from 2020, but kind of how my lab got into the field. What we were interested in is how to develop deep learning methods for histology image analysis. And so this is actually something that people do all the time now. But what you do is you take a whole slide image, might be a centimeter wide, and then you break it into little tiles. Those tiles will typically be, maybe a couple hundred microns wide. And then each of those tiles, you pass them through what's called a foundation model. Think of it sort of like ChatGPT. It breaks things down into some kind of constituent elements through this, somewhat nebulous, hard to understand neural network, which we call foundation model. And that's what's shown here. And then you train something to have some specific task. In this case, in this old paper, we were trying to predict tumor normal status of a slide or tumor subtype, and we applied it to TCGA. This is the kind of result we got. This is actually kind of a follow-up paper from that initial work. And once you kind of break down these tumors or the whole slide images into, essentially you get a vector from them by running them through a neural network. And what you're seeing here are U-maps of the kind of the vector representations of these individual tumors. And what you should see on this slide, each of these dots is an actual tumor image, but just condensed to this vector format by the neural network. And what you should be able to see is that the normal samples, there's a bunch of normal samples in TCGA, that they separate very easily from the tumor. You can just by eye see that there's a good separation. And in addition, if you map on the actual tumor types or the tissue type onto them, you can see there's a very good separation by cluster. So from this kind of neural network approach, you can build a discrimination model, which is extremely accurate to get both whether it's a tumor normal and the tissue of origin, and in fact, 97% AUC. So this just works. This is actually quite standard now. There's a huge activity to further progress these. So this is just an example. This is a slightly more sophisticated task. In fact, we collaborated with a couple of colleagues here at Yale, David Rim, and also a fellow in his lab at the time, Emily Reisenbichler. And we were interested in whether you could predict HER2 status for breast cancers. And in this case, this was an image which their team annotated. I think Emily actually did the outline here, where she just kind of circled where the tumor is in this image. And we knew ahead of time, because this is a TCGA sample, that it was not HER2 amplified. That's from the CNV status, that it's not amplified. And we could see that the neural network which we used at the time was very accurate. And first of all, predicting where the tumor is, you can see all the gray. And it also got the correct HER2 status. That's because it's gray as opposed to red. And in fact, we didn't have to take that many images to train this. This only took about 200 images. So it's not like you need like Google scale data to do this. It's really quite feasible. So this is the AUC for that process. What I just showed you is we could actually even predict trastuzumab response. So this is a 0.807 AUC, and that's, in this case on the right, that's where we used the annotation. If we kind of hadn't asked Emily to annotate it, where we just said, here's the slide, then we would have had a worse AUC. So it's not bad. This is not clinical grade, not quite what you would replace IHC HER2 with, but clearly quite informative. And this was several years ago. We haven't been working on this particular problem ourselves recently, but a lot of people have followed up on this. So that's kind of a little background. I want to tell you about something more recent we've done. And this is, again, with H&E, but to kind of set the stage for what we're doing with multiplex data. This is something that we've done together with quite a few colleagues from Yale, including Emily Christensen Legay and Shauna and Howe and others, but really a team from several different sites. This is led particularly by Jill Rubenstein, who's a senior scientist, who's a part-time surgeon, part-time computational biologist, and she works very closely with my lab. And then Adam is the grad student, MD, PhD, who worked on this. So we've been very interested in pediatric sarcoma classification because it's actually quite a hard problem pathologically. There's a very broad range of pediatric sarcomas, but they tend to be rare. And even the task for expert pathologists is not that easy because people tend not to see very many cases kind of in their own practice. And what is typically the case for classification is looking at fusions and through molecular testing, but that's still, not that, there's a cost issue with regards to that. So what we'd like to do is, what we wanted to see is if you could do this just from H&E. And so, if you can do that, any hospital could do this, even in places. Actually, we're working on a technology. I'm not going to show slides on this where we just take pictures with cell phones, and it seems to be working quite well. So I think that's something that's going to happen actually in the field very fast, and we might even spin out something. But ask me about that separately. Anyway, we built up a pretty good collection of data, and this was it took some effort because collecting these, it's still kind of a newer thing for these kinds of these rare pediatric cancers. But we got a good collection from Yale. Also worked with St. Jude's, kind of this NIH Initiative Children's Oncology Group, and also MGH. And then we had this is the distribution of subtypes that we got. So then we applied this method, very similar to what I was showing with our TCGA classification. And we wanted to know, could we classify these kind of difficult sarcoma subtypes? So this is our process. We start with the H&E images. And there's a batch effect. Different sites, they have different staining processes. But essentially, if you have enough data, then the neural networks can deal with that. Though we do have a pre-processing normalization step. We pre-process, we tile them into pieces. And then we extract image features using the deep learning models. I'm not going to go into great detail on the deep learning. Would be happy to tell people about the more technical side in individual conversations. We use, this is probably the secret sauce. I'll just say a little bit about it, but again, it's quite mathematical, so I don't want to spend a lot of time right now. But we developed a statistical pipeline, which is called Sampler, for slide level data representations. And the challenge of, one of the big challenges in digital pathology is that you kind of look at little regions at a time. And that's what I'm showing here. You break the sample into tiles, but it's not that obvious how to integrate that data afterwards. Like I think professional pathologists know how to do this. They'll say, okay, the edge of the slide, there's a freezing artifact, or there's a folding artifact, or maybe there's too much fibrotic tissue or adipose tissue. We're just going to ignore those. That's just kind of, people have that inherent wisdom. But it's not so obvious how to do that for kind of a computational method. And also you may have other behaviors where some regions of the image, they look more malignant than others. So kind of meshing all that data is not so obvious how to do it. What our sampler approach does is we do something very simple, which is that we say, the kind of relative proportions of the main behaviors are important. So for example, some tumors may be associated with fibrosis. So if I see kind of a higher fraction of fibrotic tissue nearby, then that has some information. Or if I see a high prevalence of vascularization, that's important. But we kind of put that data in a statistical sense. Okay, ask me about it separately. Anyway, we take that concept, build a representation for the slide, and then we do a little bit of more sophisticated neural networks here using a transformer. And then this FCN, the last step, is a fully connected network, and we do the subtype prediction. Again, for those not expert, from an implementation perspective, these last steps they're pretty standard now. So how did we do? So the first step was to detect rhabdomyosarcomas versus non-rhabdos. And it turns out that we can do very well with this data set. So the AUCs that we got were 0.96, really very high. We really did not expect to do this well. And this is comparable to what clinical pathologists can evaluate, kind of the best trained. And previously, the state of the art had been closer to 0.9, maybe a little bit above 0.9. But really, this worked extremely well. That's kind of the first step. Then we looked within the rhabdos, and then the two major classes there were the alveolars and the embryonal. And again, it really just worked extremely well with a 0.96 AUC. So again, we're quite happy. We then tried a more specific task, kind of a two-stage test, and that was to be able to classify Ewing sarcoma versus non-Ewing's. In this case, this is two-step. The first we do rhabdo versus non-rhabdo, and then we predict Ewing's versus non-Ewing's. The Ewing's case, we really don't have that many samples. We only had 44 total. And yet it really did a lot better than we thought. We got, in that first step for the rhabdo, in this case, it was about 0.96 AUC. And then for the Ewing, it was about 0.93 with AUC. So we're quite pleased with this, and we think that this is actually pretty close to being usable in a clinical sense. I mean, you know, IRB issues aside, it really works very well. And again, one of the surprising things I think was that you don't need that much data. Now, our approach is a little bit different. I guess I shouldn't go into this too much since I'm not really showing you the technical aspect. But our concept of how we represent the slide data, where it's essentially proportions of different types of behaviors, that actually makes a big difference. Most groups, like if you look at the machine learning literature in this field, they use something called the vision transformer. Essentially, it's a brute force strategy where they try to use all the data and all the spatial relationships of everything in the entire image. And so they just rely on the fact that there's a lot of image data. And that's how most of the kind of image AI things that you see in the public field, like things like DALL-E or stability diffusion, how people are like making movies and things like that. That's how they work. But we have a simpler concept, which we think is very suitable for a digital path. So this first section of talk, let me summarize. Very accurate, on par with manual pathology review. And because we have this special representation, actually, it's very light. We can actually do this analysis on a laptop. You don't have to do it on the cloud. And so we think this could be rolled out actually quite well, even in, you know, under-resourced settings. Yes?
Does it work in core biopsies where you need the entire resection?
In this case, we tested on the whole side image. I don't know about core biopsies. We could test that. It's an easy statistical thing. I think we might have actually looked at subsampling, how big you need. But I think there's a good chance. I mean, a lot of it is, you know, how is the actual section taken? And, you know, practicing pathologists, you know, such as yourself, that you have some intuition of how big of a piece you need. And I think the answer is if you are capturing enough for that to work, then I think this will probably work. Yeah.
No pathologist involvement at all in terms of tumor or selecting sections.
Well, we did not impose how that would work. With each of the groups, of course, pathologists are involved with their, you know, all the steps from their usual process. We did not impose what to do. We just said, please send us the samples by whatever process you guys have internally. Yes, we use that. Yeah. Okay. So now let me there's a bigger issue, I think, for the field, which is that, you know, with all these neural networks, you know, do we trust them? And it's a particularly critical issue in the clinical space. And, you know, as a scientist, the way I think about this issue is I don't really believe in something until I can poke around with it. And there's a saying, you need to be able to break something, you also need to remake it, and then you understand it. So we were thinking about this question, and can we change this from just being like, we built this neural network, you know, we believe it works, which is kind of unsatisfying. Can we actually test it? So we built something recently that allows interactive training of these spatial classifiers. And the idea is pretty simple, which is that you take these images and then you take a little piece. And then you know, a pathologist or even just a research scientist will say, okay, that little piece, do I believe it's what I'm looking for? And then just click an answer. So here you would just label it positive, negative, or maybe you don't know, or maybe you made a mistake. And then you update your classifier as you go. Okay, pretty intuitive. And so we built something which does that. And I think this is quite important because often when you're looking at these spatial images, you kind of don't know how strong your signal is. You don't know if I need to look at 1,000 slides. Can I figure out something from only 3 slides or 3 patches? You kind of need to play with it. So we built that, and I'm going to show you something. Now, there's a lot of code here. There's no exam. But the reason I showed it to you this way is because I want you to see kind of that this just how it works in real time. Because the key thing is that we built some code that works in real time, and it's based on that sampler representation, which I mentioned, which is very lightweight. So in this case, this is not a cancer problem. What we have is code which can classify fibrotic tissue in a kidney. So I'm just going to run this video and you'll see. This is code from a Jupyter notebook, and I'll stop it to make it clearer. So what we're doing right now is this is all real time. We're just loading slide data, okay? Okay, we're loading slide data, and then this, I'll pause it. The task we're gonna try to do is find this fibrotic region. It's kind of like there might be some adipose in there, but try to find those fibrotic regions. Okay, so we're just kind of the guy who made this, he's just playing around. This is in a kidney, so that's the glomerulus. So what he's gonna do is then, he's loading the data right now into the system. This will take, the whole video is about 2 minutes, you'll see. It's just loading the data. Okay, so these are a bunch, oops, I'm sorry. Let me speed that up. Okay, so now he's loading the data. And then what you should see is a bunch of tiles that come up. And okay, there it is. Now what he's gonna do is just pick one and say, that is my example tile, okay? And I can't remember if he picks which one. I think maybe this one, or it might be this one. But he takes an example that has fibrosis in it, and that's kind of the seed. And then after that, the system will kind of start prompting you with other tiles and says, just tell me if this is fibrosis or not, okay? And you'll see it in real time. Now I'll just play the rest of the video and you'll see what happens. Okay, so right now he's specifying the sample, and then this is running to say, now tell me some tiles whether they have it or not. He said that's not fibrotic. Click the negative button you see in red. This one doesn't have it, it's negative. Let me pause 1 second. Okay, so what you see is just clicking yes or no, or uncertain, and then what is on the right is our prediction. So this is the H&E on the left, on the right is the prediction, and this is whether the computer thinks that that region is positive or negative. If it's red, thinks it's not there. If it's blue, it thinks it's there. So right now, this is something that doesn't really have fibrosis, so it thinks it's all, doesn't think there's anything there. Okay, so you'll see it going from here. Okay, starting to see it. Hopefully you caught that. There was a fibrotic region. More fibrosis. It's starting to think it's there. Thinks that this is not. Okay, there's fibrosis. Okay, all that is real time. The rate at which you're seeing it is being calculated. Okay, and now what he's going to do is now he's trained a classifier. You can see only clicked about 10 examples. And then he's going to see how does that classifier apply to the entire slide. Okay, that's running again in real time, just on his laptop. So you'll see that in just 1 second. Okay, and this is what it looks like. Okay, so this is the zoom out from the whole slide image, and the blue is where it thinks along the slides, where it thinks the fibrotic regions are. And I think, I cannot prove to you those are fibrosis, but I think most of you would recognize that being on the edge of the sample seems more likely. That's where the fibrotic regions are. And so I think we came up with a pretty good classifier, really in about 1 minute. And the important thing about this is that you can just go back. If you don't believe the answer, you can try it again. Or you may say, there was a tile that I wasn't quite sure about. And so in that case, I'm going to leave it out. And so you can just play with it. And I think this is the way for us to convince ourselves that these things are working. So, okay, I'm going to go ahead. It's not maybe algorithmically obvious, the novelty, but I think it's actually quite important for our process. Okay, so as I was saying, the real-time applicability stems from this data representation. It leads to predictions that are many orders of magnitude faster than vision transformers. If we tried to do this with say, some of the other methods, the more standard methods, this would have taken many hours to run. And then we wouldn't be able to play with it in the sense and explore. Okay, so now I'm going to talk about something totally different, but circle back and tell you how it relates, which is spatial multiomics. I think this crowd probably knows a lot about this. The way I think about it is we could see in H&E 2 colors in the past. So here we can see this flower, but bees would see this quite differently. We can see these other colors, and this is particularly ultraviolet. And that's how the bees find what's important. And so now that's what we can see with our multiplex. But sometimes it's not obvious what we should be looking for. There's so many colors that that's the challenge. But there's signals that can be quite interesting. And I think in this case, we have some intuition that because it's so structured, you've got this reddish color on the right, that's probably important. Okay, so what we do in my lab is really take that very simple concept and think, if there is spatial structure, that should guide us in deciding whether something is actually relevant, not just kind of like, not only gene expression. Okay, so I'm going to tell you about a story here where we worked on triple negative breast cancer. Actually, this is also in collaboration with some colleagues at Yale. David Rim helped us out with getting some of the samples here. And the idea of this was to look at triple negative breast cancer. And in this case, we generated tissue microarray spots about 1 millimeter wide. And we profiled them by imaging mass cytometry, which is not quite as popular now, but similar to Codex, so about 1 micron resolution with a panel. I'll show you that here. These are the panel of proteins we used. And so, we kind of designed this carefully. A lot of them are immune markers, other kind of standard markers. I'm happy to take technical questions about this later. But let me tell you what we do with this data. So we developed an algorithm to analyze this. We call it Spatial, to identify structures in these multi-omic images. So the way we do this is we take the image, so it's actually a TMA spot, so it's more like a circle. We tile it, and then we extract features. So these are the protein signal from the IMC. And then we're doing a dimensionality reduction using non-negative matrix factorization. And once we do that, then the data in this kind of new representation look like this. So each of these are tissue microarray spots from this triple negative breast cancer. And the reddish colors are the tumor. The bluish colors are immune regions. And the green regions are other kinds of stromal, like typically fibrosis. So what you should see here is that, different tumor samples look quite different from another, but there is structure. Things kind of like here you've got a blob of tumor, and then here it's a little bit more sparse, kind of wispy. And again, our intuition is that the shapes that you see, that's relevant for outcome. And another thing here is that we were able to do this analysis in a way such that everything is jointly analyzed, all the samples together. So one color in one sample means the same thing as that color in another sample. It's not internally distinct. So this red is the same red here, et cetera. Okay, so then the question is, can we predict outcome from this kind of data? And so this is something, it's not exactly what I showed you, but it's conceptually similar. So in this case, there has been this idea that, kind of people have long known that there are these suppressive cells in tumors, often myeloid cells, MDSCs. And we were wondering just something very simple, which is that if you have the myeloid cells inside the tumor, is that can we predict quantitatively that that is good or bad? And so we looked at these 2 categories of these samples. In this case here, the red is the tumor, that's pan-CK, and then blue is DNA, and then green is the myeloid signal. In this case, that's just CD68. And you can see that in the left, the tumor cells are they're together, and the myeloid cells tend to be a little bit outside them. Whereas in this case, they're more highly intermixed. And so we just developed a distance metric based on that signal, and that's this total variation distance, we call it. And it turns out even with these small TMA spots, we can see that there is a signal that is associated with outcome. So, and in fact, the p-value is pretty good, less than 0.01 for those samples. We verified this in 2 independent data sets. One was from Metabrick, the other was from we call the Basel study from Bernd Bodenmiller's group in Switzerland. And we can see this, even this very simple metric with these tiny samples, it actually verifies. So we think there's a lot of information despite these samples being so small. I think the most common question I get is, well, you know, how did you accrue those TMA spots? How did the pathologist pick them? Of course, we are relying on their expertise, so it's kind of biased in that sense. But so we cannot quantify that process, I think, as well as we like. But what we can say is, according to standard practices, that groups can replicate this. So that we're quite encouraged by that. Yeah.
Density of cells, which is usually inversely related with the distance between cells measured.
Good question. I don't think we explicitly did anything for that. Probably because these are all relatively dense regions, there wasn't that much variation in it. But I don't know. We can look at that more if you think it's worth it. We also did this based on a cellular neighborhood analysis. So what you can do is, the cellular neighborhoods are kind of like what I showed you in that first picture. So we can see from that essentially it's a clustering argument, and kind of, do we see these certain color patterns, and are the prevalence of those color patterns, are they associated with outcome? Turns out that we can see some pretty clear signals from that as well. For example, if you have regions that have high tumor myeloid together and also have MX1, then if you have a lot of that together, then that's bad for outcome. Similarly, if you have a lot of vimentin with that's mixed into the tumor, we can see that's bad for outcome. And then this, I think, is really easy to understand, and we would hope this would be kind of the base case, which is that you have a tumor region and high Ki67, that's bad. Okay, I think we would expect that there would be something wrong if that were not true. So this method actually works, again, better than we expected it to. What we think is this question of how do you integrate all the spots together, and did you sample it correctly? That's kind of related to that other question I was telling you about with the sampler method. How do you aggregate all the tiles together? Something that we're thinking about on an ongoing basis. Okay, this, I think, in the interest of time, I think I'm going to skip most of this. I'll just tell you a little bit about this. We did spatial profiling of PDXs. Well, okay, maybe I'll talk about it. Okay, so we did spatial profiling of PDXs. This is melanoma. I know that melanoma, you guys have a big project here for that. And in this case, we were looking at dabrafenib, trametinib treatment, and we want to understand the spatiotemporal response. And this was led by Jill and other scientists in my lab, Sergei Dumansky. And the way we looked at this was with PDXs. So, of course, it doesn't have the immune component, but if thinking about the tumor intrinsic part of it, we could study that. And so we generated multiple mice, and then we sacked them at different times, all with the same dabrafenib, trametinib treatment. And then we profiled them by spatial transcriptomics. This was Visium at the time. And then this is just what the data look like. This is actually quite a complicated story, so I just want to give kind of the conceptual overview. So this is time on this axis, and each of these samples you see, this is actually the Visium data that came out. And so this is an untreated sample. And then if you leave it untreated for a couple weeks, this is what you get here. And you get kind of the same behavior roughly. The colors are the expression clusters. So what you tend to see is that you have this red expression pattern on the edge of the tumor. You have a different expression pattern in blue in the interior, roughly. And then these samples here are the treated samples. And this one, I'll explain this one first. It looks pink because that's the H&E, and that means there was no tumor. If it's colored, we're seeing tumor. And it's very easy for us to distinguish tumor and non-tumor because this is a PDX. That means all the tumor is human signal. And then anything non-tumor is mouse signal. So just from the expression data, it's very easy to separate. But here you can see that there was a dramatic change in the expression at an early time point of treatment. I think this is after about 2 weeks. And the tumor hasn't shrunk that much yet, but the expression pattern is quite different. And then at this time point, there's not much, you can barely detect the tumor. If you look carefully, you would see that it's mostly the same colors, these kind of orange and yellow clusters. This is when the tumor started to regrow. You can see the orange and yellow coming back. And then here at a later time point, you get, again, the orange, you get some yellow, and then the green, and then this black. We think that black cluster is actually the main resistant cluster. So there's too many things to describe here, and I kind of, this is published. Feel free to look this up. We have a lot more detail about that. But it's a very interesting data set. I'll just mention a couple of brief things, which is that, as I mentioned, that before treatment, the expression pattern looks like this. Early treatment looks like this. And there we see some very kind of like broad things, which is that these are the 2 replicates of the pre-treatment and the 2 replicates of the early treatment. And we can see, for example, that first of all, mitochondrial expression tends to be more peripheral in general, as shown in blue. But some of the pathways, they flip. They flip in terms of their central and peripheral behavior immediately with treatment. So for example, like lysosome and ribosome, they switch quite radically immediately at treatment. And then also you see like glycolysis changes very dramatically. It becomes very central as soon as you treat. And it starts off a little bit slightly, maybe more peripheral, but relatively even. Again, in some ways, these are kind of concepts that I think many of you have seen before or maybe are not surprised by, but we can quantify this very well. And we're kind of exploring these factors across more data sets as well. So this, I think I'm going to skip, sorry. Okay, so now I want to tell you how were we thinking about the multiplex data and the H&E. Okay, so the H&E is good, but the technology change is that now we can do things like Visium, we can do Xenium, et cetera. So can we incorporate that into that interactive process I was talking about earlier? So this is another of these videos. Sorry about all the code, but hopefully this will give you the right idea. So again, what we're going to do here, this is a cancer example now. And the example is we're going to try to find pancreatic metaplasia. So the data here, it'll be a combination of H&E and then also Xenium data from pancreas. And these are not samples where these are not pancreas cancer patients. These are just pancreas samples that we got. And this is part of the SunNet consortium, which my team is part of. So I will just play this and you'll see a little bit of it. Okay, so, here's the first example. What this is on the left, this is actually the Xenium data for this pancreas sample. And specifically what I'm showing here in green, that's the expression of the gene MUC5AC, which is a marker for metaplasia in pancreas cancer. And so what we would do kind of manually, I think normally when you're reviewing this kind of data, you would say, hey, that's interesting. I wonder if there are other regions which look like it. So essentially what we're doing is we're just going to look at that marker. What this program does is it will just keep bringing up tiles. And it's not random. There's actually a process where it decides interesting kind of edge cases to look at and classify. So what the user is going to do is just click positive or negative. In this case, it's quite obvious that's metaplasia, so he's going to click positive. And this is the current prediction, which is not so good. It's actually thinks the whole thing is metaplasia, but you'll see that progress over time. Okay, so now you'll see what happens. Thinks that's metaplasia. It thinks a lot of it is, but it wasn't. That one's sort of right, not metaplasia. It doesn't think it's metaplasia. So just clicking in real time, I kind of sped it up a little bit, because there were a lot of examples. And in this case, in practice, Sergey did about 50 clicks, but I'm only showing you about 20 of them. But you should be able to see that the classifier is if not linearly improving, it kind of like gets a little bit better over time. So there it thinks that the green and some other adjacent regions with metaplasia. Okay, so that one's close, but not maybe it's totally specific, but more or less gets where the metaplasia is. So now we're going to take the classifier which we trained and then map it to the entire image of the pancreas that we have, because we have a whole slide image for the Xenium data. Okay, and then this is what it looks like. So that's the mapping to the whole slide image. I think, again, I cannot prove to you that that's metaplasia, but I think that intuitively, kind of the distribution of behavior seems realistic. Like you have these pockets, and tannins, for example, are quite common in even healthy pancreas. And these ones on the left, those may be signs of that, the very small ones. This one looks like it's really becoming something that could be clinically deleterious. Okay, so that's just another example. And then lastly, I want to just talk a little bit about something that is very new. And this story is going to be a little bit maybe not incomplete, but that's part of why I wanted to share it with you all, because I know you have a lot of lung experts here, and I'm actually not a lung cancer expert. We developed this project based on some of the technology strengths at JAX. What we've been doing lately is we've been looking at tumors from aged mice. And these mice are about 2 years old. And that's something that JAX is good at. We have very good kind of mouse, we can raise them and kind of grow them for longer. So in many cases, we let them age out to 2 years. And then what we've been doing is sacrificing those mice and doing a full body necropsy and checking to see if they have tumors. So what we actually have seen is that quite a lot of them do have tumors. Depending on the genetic background, we've actually been doing kind of diversity outbred and Het3 strain mixing, if some of you know about that. And we see that almost half the mice have tumors. And the tumors vary in how malignant they are, but lung especially is the most prevalent. About a quarter of the mice have tumors of some sort. And then liver's number 2, about a quarter of those. And then the rest are kind of scattered around different types. We see a lot in spleen, and then others of different types. So we've been resecting these samples, and what we've done is started to profile them by first H&E and also Xenium. So that's it. This is that pathology process. Our general rule is we try to find regions that have a lot of tumor, but also have a boundary. We don't want to only look at the pure tumor region because we want to be able to ask questions about that interface behavior. And then this is what the Xenium data look like. Okay, so this is one example, one particular section, and this is just the clustering based on the individual cell expression. And tumor cells in red, orange is the T cells, and then the B cells in yellow. B cells over here, a lot of T cells here. This is a bronchus. This region in here, that's a lot of kind of, there's some kind of crushed lung tissue, which is there. If you were to kind of look at more carefully, you'd probably see some like kind of squashed alveoli. But you see a lot of tumors. Another thing about this is there's actually quite a lot of infiltration. It's a little staticky, but there are actually a lot of T cells in there, for example. So that's just one example. We've basically been staring at these images continuously for months and trying to figure out what they mean. Okay, so I could go on about that for hours, but this is, I'll show you some of the analyses we're doing. Okay, first thing we do is a clustering. And so this is just latent clustering on the cell expression. And the two main behaviors we see are there's this cluster 0 and the cluster 1. Cluster 0 is basically the tumor cells. And all these cells which are up here, those are generally tumor cells, but they have a little bit of range of behaviors. Kurt and I were talking about different classes of kind of lung cancers and their expression patterns. We're thinking about whether any of those correspond to different subclasses of lung cancer. But basically that's tumor up here, and then this is the non-tumor. So if you just look at those two major clusters, now I apologize, the color scheme is not the same as in the prior slide, but that's this tumor cluster, the two clusters 0 and 1, that's basically the tumor and the non-tumor is describing. So these are just some examples. You see tumor generally, in most cases it will be like quite dense, and then the non-tumor will be separate, like on the side here. This case, they're a little bit more intermixed, but they're basically, visualizing them matches up with what we'd expect based on their expression patterns. But we do see some interesting behavior, even among the tumor cell expression. All these clusters, except this yellow here, or orangish, all these are tumor. Okay, and these are actually the same color pattern as I showed you on that first image, which looked like a planet. This is tumor, this is tumor, this is tumor, this is tumor. And the clustering is on all samples at once. So that means that these are different expression patterns. So we see just a few samples that have these kind of really unusual behavior. Most of them look kind of primarily red, but these examples are different, and we don't know what they are. And as Kurt was suggesting to me, we definitely need to genotype these. We're actually, it sounds silly, but we're having some trouble with that because the samples are so small. And we've got to get that right. But this is interesting. Adam is really interested in the following question. We don't have an answer. If anyone has an idea how to do this, I would love to hear suggestions, which is, can we figure out what is the relationship of these clusters to one another? For example, here the green and the red, they co-occur. Adam seems to think that the green cells are somehow emerging evolutionarily from the red cells, but we don't know. We're thinking about that in terms of just kind of plotting these all together on that UMAP. Do we see kind of relationships? But that's not that quantitative. But anyway, that's what we're thinking about. There are definitely tumor clusters, kind of standard genes we'd expect. EpCAM, ABCA3, and KX2-1, they're in there. Kind of this top region is the tumor. We can see, if we just kind of plot differences between the stromal clusters and the tumor, we can see some of the clusters seem to be really a little bit more malignant, like this one down here, this cluster 4, has a lot of genes associated with malignancy. I'm not a lung cancer geneticist, so apologies for skimming over this. But we're very interested in this, and again, I would love to talk with those of you who are more expert about these genes, about them individually. But this cluster, we looked at also, our pathologist looked at this, and we can see that it's quite malignant. So both the mouse pathologist and our clinical pathologist, who's more expert in the human side, they both agreed that this cluster for regions, it's malignant. Okay, I mean, the others are malignant, but this is more malignant. Okay, so what about the non-tumor cell behaviors? I think I'm going to wrap up in just a second. Again, now this was the orange color I showed you before. What can we say about those? What we did was we subclustered that set. And this is what this looks like. Again, there's a lot of stories in here. I'll just say a couple of them. In this case, you saw this picture before. There's the T cells in green, the B cells in blue. And then there's definitely infiltration. This pinkish, it kind of varies. That's probably like mostly the kind of supportive original lung tissue. This sample is very interesting because we have this red cluster. Again, that's non-tumor in this case. Sorry, it's not the tumor which I showed before. And what we think it is that it's some kind of pre-malignant cell. So we don't see this in all the samples. This particular sample is very unusual because there's a lot of it. And it seems to have a low cell cycle kind of expression. So it's a mystery. Again, we don't know. Suggestions, please welcome. We also see some other things that we're trying to figure out. Again, there's more questions and answers. One is that we see this cluster here, which has notable senescence markers, which are CDKN 1A and 2A, or P16 and P21. And these are epithelial cells. I'm not showing all the markers for the epithelial at the bottom here, but those are epithelial cells. And they have a senescent signature. This cluster looks like immunosuppressive myeloid cells, has kind of classical myeloid signatures. And then this one, we're really interested in it because it has a mixture of some senescent signal, not quite as strong as the epithelial cells, but some senescent signal, and also dendritic markers. So we think that population could be really interesting to focus on more. So, okay, like I said, I cannot wrap up that story. There's too many things to talk about, but I wanted to, share with you all and hopefully get some feedback. I'll summarize by saying, image data they're really, so many things are happening. You can predict phenotypes and it in a pragmatic sense. I think as we saw for the H&E, you can actually get clinically relevant predictions pretty easily. So clearly the multiplex, it's not like we need billions of data sets. We can see things with what we have. This sampler data representation, that's the kind of the more mathematical side. Ask me about it later, but it's actually important to getting all those real-time things to work. And then some of the specifics, tumor myeloid, super interesting for actually making predictions for TNBC. And then finally, I would love to work with some of you here on analyzing these spontaneous lung cancers. We have too many directions to go in, and we need your help. So I will stop there. And these are the folks who worked on the project, especially Adam Tyson, Joe Rubenstein, Ali Fergurpour, Sergei Domansky. Lots of help from collaborators at Yale, especially for the sarcoma project, and then some others here. So thank you.
Before I forget, I just want to remind people that there's going to be a session, the Bruin Connections next door with Jeff afterwards. So if we don't get to answer your questions or you want to speak to him more directly, please join us afterwards. And then question.
Katie.
Sometimes in this person, it's not fast. So that's something that happens actually like and the fact that like I noticed at least on the what seemed like the profiles like around.
Yeah, so we can see that expression pattern very clearly. We need to get the genotyping done. We're in the process of it, and thank you for your conversation previously. We'll get that, but the expression so far looks good.
Look at signatures of that. And I think one of the really interesting things, like sort of a follow-up of this early KRAS first.
Oh, that's a great suggestion. Yeah.
Those mouse, because then that would like open the door, I think, for them a lot.
Yeah, let's talk more later, but let's make that happen. I definitely agree. Maybe here and then, yeah. We didn't do that. We should. Yes, we just haven't. Sorry. We're working through a few of the technical points, which is that it's with the Xenium data. So, oh, actually, no, I take it back. We can't do it because it's Xenium. So we don't have the splicing behavior. We do have some other samples that we are trying to figure out how to do that with kind of subsampling with a Visium HD type approach, but we don't have it for those samples. If you have a way to do it, it would be awesome to hear. Yes. Yeah.
Early talk, you talked about the distance between myeloid cells and tumor cells.
Yes.
Hypothesis at the end. And for which foreshadow better prognosis. but I think your result was the clip effect.
Oh, I might have misspoken. So I guess what we expect is that our hypothesis was that the myeloid cells, if they're infiltrated in the tumor, then it'll prevent, say, effector T cells or other effector activities from happening. And that is the effect we saw. Maybe I didn't say it clearly. The inflammation. Yeah, we do have quite a bit of surrounding region. We have not done that analysis yet. And we have some adjacent tissue that we have. It's embarrassing to say, we have not done that analysis. It's an excellent suggestion. Yeah, we could do that analysis. It's very interesting because we have the whole body. So, right? So if there are other regions to look at where that would be particularly prone to joint inflammation.
Find that many metabolic inferences could be very interesting if you're very forward.
Yeah.
If people work in cutting transcriptional affiliates.
Yeah, that's a great suggestion. Thank you.